This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 811, recorded on September 30th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York, Daniel Griffin.
Hello, everyone.
And I hesitated slightly because it's the end of September, Daniel. And this is update
30 days has September, April, June, and November.
Boy, it went by quickly, didn't it?
It did, actually. How's
the pandemic looking from your end, Daniel?
You know, it's really tough. I was actually thinking about this just over this last hour. There's always the numbers. They're like, what are the numbers? What are the waves? But for me, a lot of COVID is sort of personal, right? I mean, and so, direct patient experience, having patients die, having patients not do well. And that continues. I have a woman in her 40s, unvaccinated, in the ICU right now, and we're talking about, is she going to get better? Is she going to end up on ECMO? And, tragedies like that. I'll share this story because I think it's illuminating. This is a family friend, has a niece. The niece has Down syndrome, and the parents are anti-vaxxers. And I won't say vaccine hesitant, I'm just going to say full-fledged. So the wife had to get vaccinated for work, but the father didn't. And the father, about 2 weeks ago, actually got infected with COVID. And what often happens, he got better. And this then reinforced his ideas that it was so much better to get infected with COVID. Why get vaccinated? And there was a lot of discussion about, well, what about your adolescent daughter? With Down syndrome. Well, what about her? I mean, she's at risk. You haven't vaccinated her. And I think we know where this goes. The adolescent daughter in her late teens ended up getting infected with COVID, had a positive test, reached out to the pediatrician, encouraging monoclonals, right? First week. The pediatrician was not comfortable with the data on monoclonals and decided not to. So this young lady ended up in the hospital on oxygen. And when she went into the hospital, they noticed that her lymphocytes were low. So they were concerned that maybe her lymphocytes were low because she coincidentally also had leukemia. And so they didn't want to start steroids until they had a chance for a hematology consult, which did not occur until Monday. She admitted Friday, hematology consult on Monday. Finally, Monday evening, they decide to start the steroids. By Tuesday, she's in the ICU on high flow oxygen. She's required CPAP. Not doing well. I mean, this is all, vaccine preventable. And I always tell the stories. Like, I don't tell the stories about when everything goes smoothly. I think we learn from lessons like this. So, unvaccinated, putting a family member at risk, and maybe our audience isn't aware of, Down syndrome, about 4 times more likely to end up in the hospital, maybe 10 times as likely to die. I mean, so this is a high-risk condition. This young lady, a little bit heavier than would be ideal, also putting her at increased risk, not getting the monoclonals, not getting the steroids with that delay, and then sort of question about, should she be given Tocilizumab? And I'm going to get to some data on that. But by the time you're, requiring CPAP and high flow oxygen, you probably missed the window. We're going to make a benefit there. So for me, the pandemic is still continuing to be incredibly painful on a personal level. But looking around the country, we are seeing a little bit of light. We are seeing, that things in many areas are getting better, but still an unacceptable high rate of infection in our younger population.
Very sad story.
Yeah, it's just... All right, let me start off with our quotation. Knowledge is no guarantee of good behavior, but ignorance is a virtual guarantee of bad behavior. And that's by Martha Nussbaum. I love that. And hopefully what we're doing is we're providing education. I was doing a talk for one of the California schools last week, and sort of the final question was one of those. There's no right answer to this, but it was about vaccine hesitancy and having conversations. And I guess my view has always been the long game. If you are respectful, if you continue to provide education, I think that you may not get people to make the right decision now, but you're building toward better decisions, better behavior in the future. So, all right, children and COVID. I like to say children are at risk for COVID. There's no doubt. I think this is very clear. And also wearing a mask is less traumatic for a child than being hospitalized. And I think our story reflects on that. I was on a call earlier this morning with the consortium that I talked about, where we're really trying to get good data on COVID in children, and also whether or not, as we've moved into the Delta variant, whether or not that is somehow having a different impact. But one of the things that came sort of to my attention on the conversation is something we've talked about a little bit. There's this idea that, maybe this will become the next endemic coronavirus. Maybe children, if they get infected in the first few years of life, they'll have this immunity that protects them going forward. But one of the things that was shared, and this is consistent with what we're seeing here in the US, if you look at under 18 and you actually look at 0 to 4, right? So babies and really young children, that actually has for a while now had the highest associated rate per 100,000 population of ending up in hospital. So this whole idea that it's a continuum, the younger you are, the better you're off with COVID, actually breaks down in the 0 to 4. And one of the gentlemen on the calls was from Brazil, and he was actually saying this has been a huge problem, really, particularly that first year of life. They're seeing a lot of hospitalizations, they're seeing deaths, and so sort of reflecting a situation. Vincent, did you have a thought on that?
Yeah, just one condition that might mitigate this. If in the future, when most mothers are immune, they will pass antibodies to their babies, and that may reduce severity, and then the baby is somewhat immune and it reduces disease subsequently.
Yeah, I like that idea. And that's interesting because I know with Dixon, when we've been on this week in parasitism, we've talked about when do we see the mortality for childhood malaria, and it's after the children have lost that protective antibodies from mom. So I hopefully that is an optimistic, and hopefully that will pan out, because this is, if you think about it, what an incredible challenge to think that we're going to continue to be able to keep our population. Well, we haven't even gotten there the first time, but the idea that we're going to be able to always vaccinate against SARS-CoV-2. But now I'm going to say in the child world, maybe this is the most exciting news for this week's. Is on Tuesday, September 28th, Pfizer and BioNTech submitted initial data to the US FDA from their pivotal trial on COVID-19 vaccine in children 5 to 12 years of age. And then they went on to say, and I think this is really important so that people have the clear message, formal submission to request emergency use authorization to follow in the coming weeks. So the clock is not really officially started, right? And that clock is when they request EUA, there's a 15-day period of public comment, and then we hear from the FDA if they're going to extend it. So we are all keeping our fingers crossed that there will be vaccines down to 5 years of age by Halloween. And I guess that moves us right into active vaccination, right? Vaccines, the jabs that keep you from getting sick. And this will be a public service announcement right here. It's time to get that flu shot, right? We're by the time this drops, we're in October. And the current guidance from the CDC for the use of COVID-19 vaccines is that these vaccines can be co-administered with other vaccines. So COVID-19 vaccines, influenza vaccines, you can co-administer. Early on, we were spacing. In part, we were trying to understand the reactogenicity, things like that. At this point, don't miss an opportunity. Never miss an opportunity to vaccinate. If the person comes in and they want to get a COVID and a flu shot, we can do both at the same time. Well, how are we doing with the third dose? We had the early release from the MMWR safety monitoring of an additional dose of COVID-19 vaccine in the United States, August 12th through September 19, 2021. And basically, the adverse reactions, the reactogenicity that we're seeing with dose 3 is really similar to what we saw with dose 2. So during August 12th through September 19, 2021, among 12,591 VSafe registrants who completed a health check-in survey after all 3 doses of an mRNA COVID-19 vaccine, 79.4 and 74.1 reported local or systemic reactions, respectively. After the 3rd dose, 77.6 and 76.5 reported local or systemic reactions after the dose. So this is really very, very similar. We're seeing about the same. We know that now over 500,000 folks have jumped in now that there's a booster, but we knew over 1 million had done it ahead of time. But I think this is important. We've still discussed a little bit about unknown benefit, but really critical was following and continue to follow safety data. And so far, voluntary reports have found no unexpected patterns of adverse reaction from this additional dose. The CDC is going to continue to monitor the vaccine safety for these additional doses as they go out. Now, one of the things I want to talk about in the vaccine area here is one of these anti-vaccine tropes that's going around. It seemed like there was a lot of it this last week as healthcare worker vaccine mandates were going into effect. And this was this whole idea that, I've already had COVID. Why are they forcing me? Why are they making me get vaccinated? If you already have COVID, I'm never going to get this again. And this is this whole idea. There's a local ID doc, and he always just seems to muse about, I don't understand how the vaccines can be better than just getting infected with COVID. And most of us roll our eyes and wonder if he's ever seen a second case of dengue or he's ever seen someone with tetanus and, where this comes from. But Oklahoma now has its weekly COVID-19 report, including reinfection data. I thought this was really interesting. It's actually got quite a little bit of press, but they now are reporting since May on the reinfection rate per 100,000 eligible cases. And you can see this really has started to march up from 358 to 512 up to now in September, over 1,000 per 100,000 are getting reinfected. So I think it makes sense and it matches experience. That first 3 months after an infection, you're at a low reinfection rate, but it does really seem like that protection drops off. This is, I have to say, there's 2 aspects. So one is it's a little bit frustrating because I would like there to be better protection against reinfection. We do know that there is a benefit to getting vaccinated, and we know that if you've been infected and then you go ahead and get that vaccination, you have dropped your risk of reinfection by about 2.34 in that nice study from the MMWR. So we are routinely asking people not to have this musing and just everyone go ahead, get vaccinated. You have any comments there, Vincent? From the incubator in Manhattan?
Yeah, that's where we are. We're at the incubator now, recording everything here. And these are all cases in people who are not vaccinated but had previously recovered from COVID. Is that correct?
Yes.
And what is the overall in Oklahoma frequency again?
So what they're looking here is at the number of cases. It's funny, they call it cases eligible for reinfection. So these are individuals who had a prior infection and now looking at the reinfection cases and then counting them up over time. The test positivity rate in Oklahoma is about 15%. So they have actually a very high test positivity rate. So here you had 453,927 eligible for reinfection, and they had 1,035 that ended up reinfected.
What's that rate? Do you know offhand?
So that's the rate per 100,000. So you could actually multiply that by 4.5. So it really was over 4,500 reinfections just in September.
So can we compare that to vaccinated people?
Yeah, they do that. They compare it. And we see a range, depending on when you look, of somewhere between 2 to 9.5 times as likely to get infected compared to the vaccinated people.
Okay, good. That's good. I wish more states did that.
Yeah, actually, it would be nice. It would be nice. All right. passive vaccination, right? We talked a little bit about monoclonals in our opening story. On September 29th, so just yesterday, the article, just yesterday when we're recording, it'll be a few days for folks listening, the article Regencove, Antibody Combination and Outcomes in Outpatients with COVID-19. This article came out as a peer-reviewed publication in the New England Journal of Medicine. And this paper describes the phase 3 portion of an adaptive trial where outpatients with COVID-19 and risk factors for severe disease were randomly assigned to receive various doses of intravenous Regencove or placebo. So we've talked about a little of this data in the early preprint and in the press releases, but in the original phase 3 portion of the trial, 3,088 patients with or without risk factors for severe COVID-19 were randomly assigned to receive a single intravenous dose of Regencove. And it's either 8,000 milligrams or 2,400 milligrams. And the difference in dose, think about important in part for volume, right? Because we're also thinking the potential that we can deliver these subcutaneous or IM in some. So in the amended phase 3 portion of the trial, there was an additional 2,519 patients. So they're sort of growing the numbers here. The median follow-up was 45 days. They're extending the follow-up from that initial 29 days. 96.6% of the patients got past that 28 days and had this longer follow-up. Now, this is, I think, critical because timing, and I keep getting at this, these patients had a confirmed SARS-CoV-2 positive diagnostic test no more than 72 hours prior to randomization and onset of any COVID-19 symptom no more than 7 days before randomization. And they found that the COVID-19 related hospitalization or death from any cause, there was a relative risk reduction of 70.4%, p-value 0.02, with receiving the MAB, the monoclonal antibody therapy. The median time to resolution of symptoms was 4 days shorter in the folks that got the Regencove. And this was similar at the two different doses. Serious adverse events, right? People are, I'm worried about giving people this. 4% in the placebo group, 1.1 and 1.3% in the two dosing groups for the MABs. Infusion-related reactions occurred in less than 0.3% of the patients in all groups. So this, just to reinforce, this is a very safe. Your patient is much more likely to have problems not getting the monoclonals than they are getting the monoclonals. And the monoclonals continue to really do a tremendous job of reducing the risk of COVID-19 related hospitalizations or death. This really still remains our most effective antiviral therapeutic. But what about the real antivirals, right? The traditional, where are the pills? And so on September 27th, Pfizer announced that they were starting their EPIC-PEP trial. So that's evaluation of protease inhibition for COVID-19 in post-exposure prophylaxis. So this is really interesting, sort of moving on to the second level. We know we have a number of trials going on where people get acutely ill. We try to rapidly give them antivirals. But now we're already moving on to this study where they're looking at a household. So this study will evaluate the novel protease inhibitor PF 073213332, co-administered with a low dose of ritonavir. That's actually a pharmacological boost, so it keeps the drug from being metabolized, so it can be present and effective for a longer period of time, something we learned from the HIV days, for prevention of illness in adults living in the same household as someone with COVID-19. So the whole idea here is someone in the household, you're at high risk, you give it to everyone as a post-exposure prophylaxis. And this can potentially be administered at the first sign of infection, or in this case, exposure to SARS-CoV-2 without requiring the person to be hospitalized, without requiring intravenous therapy. So we're looking forward to that. This will be a phase 2-3 trial. And this is going to be twice daily for 5 or 10 days. So we'll get some information on how well this works. All right, the period of detectable viral replication, right? And hopefully we keep through repetition, sort of reinforcing what are the evidence-based interventions at each stage, right? So here, now we have the period of detectable viral replication, the time for monitoring in monoclonals, not the time for antibiotics. They are not helpful, potentially harmful, not the time for steroids. Very clear that we can do harm. But the monoclonals have now been added to the WHO COVID living guidelines. So the WHO now recommends antibody treatment for COVID patients at high risk for hospital admission and for severely ill patients who have no natural antibodies to COVID-19. So that's a little bit different. So we'll go into this a little bit. So what they have to say is a treatment combining two antibodies, and here it's the Regencove, so the Tocilizumab and imdevimab is recommended by two specific groups of patients with COVID-19. This is the WHO Guideline Development Group, which is a panel of international experts and patients. And this was in the BMJ on Friday, September 24th. Now, the first group are patients at high risk of hospitalization. But the second group, these are individuals who actually have severe or critical COVID-19. These are those individuals which actually we're getting into the next group. They're getting hospitalized. They're starting to get into that early inflammatory phase, but they're still seronegative. They still do not have measurable antibodies. And it's the second recommendation that is actually outside the current EUA in the US. So the second recommendation, so giving monoclonals to severe or critically ill COVID-19 patients who are admitted but still are seronegative, is based on the data that we discussed a while back, became available in June, the recovery trial, where we saw that Tocilizumab and imdevimab reduced deaths, and they actually gave a range. They suggested that 49 fewer per thousand, maybe as high as 78, maybe 87 fewer in the critically ill, also reducing the need for mechanical ventilation. So we'll have to see when that gets updated here in the US, but WHO is actually recommending not just that first week, but if you check serology data on admission, this is a group that we have data suggesting that they would benefit. So still outside the EUA here in the US. We have to wait for that. We're also anxiously waiting for the data, as I mentioned, the other oral antiviral. So we have Merck and Ridgeback Biotherapeutics with molnupiravir. We have Roche and Atea Pharmaceuticals with AT527, and Pfizer's drug, as we discussed. It's actually interesting. There's a little bit of a challenge to recruit for these trials. So hopefully we can get those trials done and we can get that information. All right, let's move into the early inflammatory phase. This is usually when people get hospitalized. And we're going to be talking about tocilizumab again. And really hitting, as we keep hitting on, how critical timing is for different therapeutics. So in the Journal of Respiratory Care, the peer-reviewed publication Impact of Timing of Tocilizumab Use in Hospitalized Patients with SARS-CoV-2 Infection was published. And we've talked previously about the 8 randomized controlled trials, including the recovery trial again, that demonstrated a mortality benefit for patients and led to the inclusion of tocilizumab in the IDSA guidelines, the NIH guidelines, the WHO COVID-19 guidelines. Now, several healthcare systems, unfortunately, as tocilizumab has been embraced, there's been some degree of rationing. So several healthcare systems have tried to create guidelines so that everyone doesn't get tocilizumab, but hopefully that it's going to the patients that can benefit the most. We'll see if that's working out. And one of the rationings has been to wait until patients are on high-flow nasal cannula or higher levels of oxygen support before they get tocilizumab. We're going to see from this trial that maybe that is waiting too long. Maybe that is after the window of benefit has closed. So the authors performed a retrospective chart review of 11,512 patients infected with SARS-CoV-2 who were admitted to a New York health system from March to May 2020. I will mention that healthcare system's Northwell. I actually know the authors on this paper. Tocilizumab was administered to subjects at the nasal cannula level of oxygen support to maintain an oxygen saturation of greater than 88%. The primary outcome of interest was hospital mortality, and the secondary outcomes were progression to mechanical ventilation, prevalence of venous thromboembolism, renal failure, change in C-reactive protein, D-dimer and ferritin levels. Propensity score matching was used to minimize any selection bias. And what did they find? Let's cut to the chase here. The hospital mortality was significantly reduced in the Tocilizumab group when it was administered at the nasal cannula level. So this was a drop from 22% down to 10.4%. So a 53% mortality reduction when Tocilizumab was administered at the nasal cannula level. If they waited till the individuals were requiring non-rebreather, high-flow nasal cannula, non-invasive ventilator, you actually were losing any benefit. So the window basically was closed. So we're hoping we'll get our guidelines sort of updated with this. And so just a little shout out to the corresponding and lead author, Anup K. Singh. He actually sent me this sort of when it was still in the galley proof stage. So I got to see all the corrections. But he's really been, I think, a leader. The folks at the Lenox Hill Hospital in the city have really done a good job of sort of understanding the disease, really being on the forefront. And so I think this was helpful. But just to reinforce, timing really matters. If you give it in that first several days, there's actually a wonderful figure, this figure 1, where not only can you see the mortality benefit relative to what level of oxygen support they give, but you can also see how important the timing was. If they got the Tocilizumab within the first couple days, even out to about day 5 with the nasal cannula, you were seeing low mortality. But once you waited, started giving it after day 5, you really see that mortality start to go up. So earlier at the right time, people who have maybe gone from 2 to 6 liters of nasal cannula, who have inflammatory markers, who are progressing despite steroids. But if you wait till they're on high flow nasal cannula, you've waited too long, and you're actually giving the medicine to people that don't benefit from it.
What does it mean, nasal cannula? Is that nebulized?
No, actually, this is great, right? We've all seen the movies, where the person has this plastic thing and they've got little prongs in the nose. So that's nasal cannula. And actually, I appreciate jumping in here because let's talk about oxygen therapy and what I mean here. One of the most important things for us as clinicians treating patients with COVID-19 is what level of pulmonary support do they require? So one of the first things I want to know when a patient shows up in the hospital is what is their level of oxygen saturation on room air. So I'll often turn off the oxygen because I want that number because I want to know. I mean, we had a patient yesterday came in, he was on 3 liters of oxygen via that nasal cannula, sitting in the 90s, but then we turned it off and he was still 95% with it turned off. So you really want to get that baseline. Now, the next thing is once you establish that they do require oxygen, so they do have a level of hypoxemia that warrants admission for severe COVID and oxygen support, how much do they require? Now, that nasal cannula that we're used to seeing in the TV shows can deliver, we say up to about 6 liters. Sometimes we'll turn it up to 10. And then the next level of support is something that we call a Venturi mask, which allows us with a twist of a knob to deliver a slightly higher amount of oxygen that we can regulate. And then we move to the full non-rebreather, which is the mask with the little bag below it, sort of reminiscent of what you see on an airplane. Above that, something we're using more and more is a high-flow nasal cannula, where instead of the small prongs, you actually have 2 larger prongs that really fit tightly in the nares, and we might deliver 30, 40 liters of oxygen per minute directly in through that device. Sometimes we'll even put a non-rebreather on top. The next escalation is to the continuous positive airway pressure, the biphasic positive airway pressure, where there's a different force in versus out. And then often we have to move on to mechanical ventilation.
So how do you get the tosi in the nasal cannula?
Oh, so the tosi is intravenous. It's just going in the vein. So when the person, gets to, they've gone from 2 to 6 liters, we've already given them steroids, you administer the Tocilizumab as an intravenous medication.
So when they say at the nasal cannula level, it means that's where they're getting oxygen, not...
Yes.
Yeah, okay, got it.
Not in their nose. Don't squirt it in their nose, listeners.
Okay.
Okay. All right. And I think that's critical. Just reinforcing timing really matters with this disease. And all right, I'm going to try to keep us from going too long today because I think we've been getting a little bit long. I know it's helping some people's fitness level. And the dogs are liking me. But long COVID is not just a 2-week viral illness for many people. And apparently we need to keep reminding people of this because I've actually started to see literature now, again, sort of this return to dismissiveness. But the paper, Incidence, Co-occurrence, and Evolution of Long COVID Features, a 6-month retrospective cohort study of 273,618 survivors of COVID-19, was published in PLOS Medicine. And here, this was an electronic health record study looking at data from 81 million patients. Included in this were those 273,618 COVID-19 survivors. And they looked at the incidence and co-occurrence within 6 months and in the 3 to 6 months for 9 core features of long COVID. And basically, just to reinforce, again, in this really large data set, they found that COVID-19 was associated with long-term effects that were common and diverse, with 57% of patients having at least one long COVID feature recorded in the 180 days after infection, 37% still having issues 90 to 180 days after diagnosis. And I'm going to close this out here with no one is safe until everyone is safe. And I'm going to give a shout out to TWiV 807. I like all the TWiVs, but I really enjoyed the shot Yearned Round the World with Chatty Sadroy and Caroline Wagner. Really nice discussion about what would have happened in different scenarios where vaccines got out to other parts of the world where there's a little more vaccine equity. And along those lines, with that theme, I'm going to remind everyone, go to parasiteswithoutborders.com during the months of August, September, October. Click that donate money and help us. Click that donate button and help us support floating doctors. They're going to be working with the Ministry of Health down in Panama, getting those vaccines out, continuing to provide care down there. And I have to say thank you so much because our listeners have been incredibly generous. And we need the funds to do what we do. These organizations need the funds to do what they're going to do. Be part of the solution. So thank you so much.
Time for some email questions for Daniel. You can send yours to daniel@microbe.tv. Ronald writes, I'm writing from Florida where the sheriff's department bans masks, won't let you in if you're wearing one, and the governor is bent on endangering the lives of our children. I know you've discussed ivermectin ad nauseum. I'm hoping you could take a look at a site that exists solely to promote ivermectin for the treatment of COVID-19 and give me some tips on how to debunk it. IVNmeta.com is a site devoted solely to convincing people that ivermectin is the cure. It spends a good bit of space listing countries that use it. The main method is to use web crawlers to find peer-reviewed studies on all sorts of sites. And they mention straight out multiple treatments are typically used in combination, including monoclonals. I've pointed out to those citing this site as science that a treatment of tap water and monoclonals, if given early, will show benefit, not all due to the tap water. I showed them your last assessment, and the response was, I don't know why the fine doctor says there's not enough evidence. There's tons of proof, and they point to the site. It appears that they find any study that mentions ivermectin, web scrape, the most serious outcome, the count of participants, and the percentage benefit, do something they call exclusion based on sensitivity, and then add the counts and percentages to their total to give them one huge study that has supposedly meaningful adults.
Yes. Okay. I was at the end there. I was reminded of Mark Crislap's. He always talks about when you take a whole bunch of cow dung and you pile it high enough, suddenly it turns into gold, and that doesn't work. You've got to look at each little bit of cow dung, and if it's cow dung, you throw it out. You don't pile it on top of each other. I think hopefully we're going to learn from the ivermectin story that if you leave a vacuum, things like this happen. We needed to do proper randomized controlled trials in this country to assess whether or not there is or is not a role of ivermectin. We haven't done those. There's been the Egypt study that never happened, the Peruvian study that seems to have never happened. There's meta-analyses that used those studies that have not been retracted. I think this has really become a difficult situation. I think that Bill Gates was really smart early on to put the microchips in the ivermectin horse paste. So, he's accomplishing his microchip control that way because he knew those people would not take I'm joking. But no, this is really challenging. There are clearly people out there who cherry pick to support their conclusion. Science is not about supporting your conclusion. Science is about discovering the truth. And the way you discover the truth is by being honest, not looking for support for your agenda, but looking for reliable data that can allow you to find the truth. And so, this is very challenging.
John writes, are there numbers anywhere for the vaccination rate among those who have already had COVID? Does anyone who has already been through it refuse a jab or two? I suspect the percent is higher than overall anyway, which might be another argument to the stridently obtuse.
Yeah, I don't, we're starting to get, as we talked this week, about a little more granular data. And I do not know, where to get really good numbers on the breakdown. I've seen numbers that break it down by political party, which I'm not that interested in, but I would definitely be interested in this. But, there is, I'm going to call this myth that there's just these hordes of anti-vaxxers who won't get vaccinated. Most of us are getting vaccinated. The majority of people who are eligible in this country have gotten vaccinated. And with the mandates, some people that we're not that excited about have either gotten vaccinated because they had to, or they have basically been given a way to save face and get vaccinated. But no, this would be interesting to see what those numbers are. Hopefully what will happen here is that anti-vax trope about natural immunity being so wonderful will be dispelled, and people will clearly see that growing numbers of individuals previously infected are getting reinfected. And the data we already have is that vaccination, even in a population that was previously infected, can significantly decrease your chance of getting infected again and having a bad outcome.
Nick writes, my wife is pregnant, 24 weeks. She had COVID in January this year. We've read articles stating that immunity given after infection is better than the immunity given after vaccination. So based on that, she doesn't want to get vaccinated until after she gives birth. I'm an ICU nurse, fully vaccinated, who deals with COVID on each shift, and I'm concerned given how I see the worst of the worst with COVID.
Yeah, no, I'm going to share your concern, and what are the things that we know here? One, and we just touched on it a little bit, is people who like the idea that natural immunity is better than the vaccine do cherry pick data. There was a preprint, the famous Israeli preprint that suggested that natural immunity prior infection was better than vaccine, has been touted and circulated out there. It's not peer reviewed. It's not reliable. We actually talked about some state level data in Oklahoma suggesting that vaccinated people are better off, are better protected. We also know that if you were infected before and get a vaccine, you're going to more than 2.3, more than twice as likely decrease your risk of an infection. And remember, a pregnant woman is trying to make a decision for herself and for the newborn child. Pregnant women are at much higher risk of ending up in the hospital. They're at much higher risk of ending up in the ICU and on a ventilator if they end up in the hospital, and they're significantly more likely to die. So this is a very high risk period. This is not a great time to be relying on natural previous infection immunity when you can have that extra safety of getting a vaccine. And we also know incredibly safe, no safety signal. So there's no reason not to get that extra protection if you're a pregnant woman.
And finally, Jessica writes, as a young nursing student, I was taught that when giving injections that were not meant to be IV, care should be taken after inserting the needle to pull back and make sure no blood came back. Now retired, I've been told most people don't bother with that anymore. And then she says, what do you think about this study where mRNA vaccines were injected into mice, one group intravenously, one group intramuscularly? They removed heart tissue, and they found that intravenous injection leads to acute myopericarditis in mice. And she says, it's only one paper, it's mice, but what do you think?
Yeah, no. So I feel like we're circling back, which is interesting. I'm old. I was trained, put the needle in, pull back. But then they did a number of studies showing, no obvious benefit and actually an increase. Actually, it's painful. Actually, apparently, when you pull back like that. So the recommendation is currently a couple things. One is don't pull back on the plunger. The other is if you're going to not be doing the deltoid, to do the lateral thigh instead of the buttocks. So things have evolved over time, but this is interesting with the mRNA vaccines. Would we be better off returning to that pullback, the plunger? I don't know, but it is worth studying, because even though the incidence of myocarditis is incredibly low and most cases tend to be mild, I think that's important to point out. A myocarditis that lasts 48 hours resolves with, now we're months out, not seeing any sequelae, a good resolution. It's quite different than the myocarditis that you get with COVID. But still, anything we can do to reduce that risk, I think is worth studying. So I would welcome. I mean, that's what we have to do. We have to keep studying. We have to put more money into the science, because we certainly see how expensive it is to not put the money in the science.
That's COVID-19 Clinical Update number 82 with Dr. Daniel Griffin. Thank you, Daniel.
Thank you, Vincent. Everyone, be safe.